Hello, this is Stephane Bancel. First of all, let me tell you I'm very thankful for having the chance to address this group. And as we look forward to using the mRNA technology, I will answer some of your questions. As some of you can guess, since we started the company 10 years ago, we believe that if mRNA could work for one disease, it could work for a lot of diseases because of the information nature of the mRNA molecule. So while we have had some good success with a COVID-19 vaccine, and we have a lot of infectious disease vaccine in the pipeline today, we are also working on other applications. So let me maybe answer some of the questions I got. So I got the first question from Jeremy Reiter around what do I think about the chance that our vaccine against cancer will work. So I would say this is, Jeremy, couple $1 billion question. We of course believe there's a chance. If not, we will not have invested in the science and run the clinical study. But as you know, in oncology, one wants to be very, very humble about the complexity of a biology and the different disease state of every patient. What we will have is at the end of this year, in the fourth quarter, clinical data in a head-to-head study in melanoma setting, where we will have patients that will have only received Keytruda and patients that will have received Keytruda plus Moderna personalized cancer vaccine. So for those of you that are not aware of this program, it's basically a super cool program where we take a biopsy of somebody who has cancer. We next-gen sequence the entire genome of that cancer cell. We do the same thing of a healthy cell of that person. We send both to the AWS at Amazon. We look at all the mutation, and then we have developed an algorithm that actually is learning through the study to identify 35 neoantigen of that patient that basically goes down into our factory in Norwood, Massachusetts, the same factory where the COVID-19 vaccine that many of you got is made. And we make in a big robot that the team designed that look like a bit like a big American fridge. We make a product for one human being at a time. It takes a couple of weeks, and then we send it back to the clinical trial site to be injected as an intramuscular. And so the idea here is, could you mobilize and help the T cells by using those neoantigen and get a better response than Keytruda monotherapy? So it's a head-to-head study. It's powered. So Jeremy, in the fall, we should have a better sense. The second question I got was from Regis Kelly, which is a technical question around can we, coat the formulation or delivery mechanism of our mRNA to get it into different cells and to target specific cells. So as of today, Regis, we're not able to do that with a delivery system to go, let's say, only in hepatocyte of the liver or only in Kupffer cell of the liver. So the formulations we have developed go to different cell type. But what we can do, which is very cool with messenger RNA that you cannot do with a small molecule or large molecule, is we know how to turn off the mRNA molecule that contains the instruction of a protein we want to make as a drug. We can turn it off if it's in the wrong cells using microRNAs, a complementary sequence in the 3' UTR region of the mRNA molecule that we design. And so let's say if we have a delivery system that delivers the mRNA into Kupffer cell and hepatocyte, but we only want for that drug expression in the hepatocyte, we can code in the 3' region of UTR of the mRNA the complementary region to the microRNA of the Kupffer cell. Like this, when the mRNA will be delivered into the Kupffer cell, it will hybridize to those microRNAs and will not produce a protein in the mRNA of our drug. But the mRNA that is delivered into the cell next door into hepatocyte will produce the protein on demand. And so that's how we can regulate very high specificity. We use that technique a lot, for example, in our cancer program, where not like the personalized cancer vaccine we just spoke about, but into a program where we inject directly into tumors, cytokines. We want to create inflammation in the tumor milieu to try to improve the efficacy of existing drugs like Keytruda. And so in that case, what we did is we coded some of the microRNAs so that as the product leaks into the body, we know the couple of cells where it's going into in addition to cancer cells, and we have turned it off so it does not produce the cytokine in those cells. So that's a pretty cool feature of the technology. And then I had a third question from Christopher Carlson, which is really around what else can you do. So we talked about cancer. Another piece I'm very excited about is, of course, rare genetic disease. We have quite a number of rare genetic disease drugs that target the hepatocyte in the liver, where, as you know, the liver being a protein factory, there's a lot of protein made in the liver, and the function of the liver makes it that kids have rare genetic disease like MMA, methylmalonic acidemia, PA, propionic acidemia, and many more that are not addressed today, for which we have drugs in the clinic, dosing in patients right now, where we basically get through an IV injection the drug into the livers, in that case the hepatocyte, to give the protein that those kids are missing because they get, of course, the wrong genetic information from mom and dad. So we are hopeful that we should get data this year in 2022, and if this works well, because the biology is very well understood for rare genetic disease, unlike in cancer, to go back to the first question we had. And so we are preparing a very large pipeline of drugs in the liver. We're also going to go most probably this year into the clinic, delivering mRNA inhaled into the lungs, working with Vertex on cystic fibrosis. So it's still a rare genetic disease, but delivered into the lung this time. We're also working in cardiology, where AZ, AstraZeneca, has a phase 2 program right now, injecting mRNA coding for VEGF into the heart for after a heart attack to revascularize the damaged part of the heart. We are also working into autoimmune disease. And so over time, we really believe Moderna has the ability to go after a large number of diseases that cannot be addressed with small molecule or large molecule. So we're very excited about the technology and what it can do over time for patients. So with this, I hope that was helpful. I look forward to engaging with some of you in the weeks and months to come. Thank you. Have a great meeting. Bye-bye.